So I was talking about some of the challenges we've had we've been focused on a pediatric brain tumor. And the field's really exploded in terms of sequencing technology, specifically in cancer, and our ability to identify genes that are involved in the disease process with the sort of motivation that we can pair these with the drugs. And I think those are ongoing efforts right now in clinical trials. Sort of personalized medicine, personalized medicine approaches. And we applied this to a pediatric brain tumor type called ependymoma. And I'll take a step back first and acknowledge that what we're finding that's distinct in pediatric brain tumors is that they have a much lower mutational rate than adult tumors, which makes it challenging to identify targets for therapy. And this is one case in a tumor called ependymoma. It happens in young infants. It's a highly lethal brain tumor. The treatment is surgery and radiation. And then when these tumors recur, which they often do in infants, we re-irradiate and we re-irradiate. And so you can imagine the long-term neurological side effects of that. So we have no target or drug that we can recommend for these patients at the moment. So we thought that genome sequencing would be our answer to that we'd identify the hits and then these would pair us with a pathway and a drug, and we'd be able to test something in clinical trials for patients. We were disappointed though, in that this didn't fit the textbook kind of approach for many adult cancers, and it was clear to us that we needed to utilize more different approaches, sort of looking less at the sequence of the DNA, but more about how the DNA folds and what regulates the genes that are transcribed from the DNA. And that's really an advancements in genomic technologies that have allowed us to perform. This is sort of a field called epigenetics, where we're looking at or transcriptional regulation, where we're looking at sort of the proteins that regulate how high is the genes expressed and sort of the folding of chromatin. And this really mapping across the genome is giving us a new paradigm, because it's allowed us to identify pathways that we never would have thought of using more traditional approaches. And we show that, at least proof of principle, that we can pair this with specific drug targeting strategies. So we think this is a new paradigm that is complementary with more traditional approaches, which could be effective in many pediatric cancers that have very low mutational rates, or even adult cancers where we found the actual driver event that we know causes the disease, but we've yet to identify a drug that we can pair with it. So I think one of the limitations is that, at least in our system, is the models. We're learning about the disease faster than we ever have. One of the major challenges is trying to develop animal models that we can test, because so we identified a drug and we tested in one animal model. We'd like to expand it to more mouse models to be able to have solid evidence that this might be an effective target we can bring into clinical trials, because we've identified a lot of targets, and I think just coming down to our most promising one and then validating that in mice would be the evidence we need to take that to clinical trials, because at least in North America right now, there are no agents that we can recommend for clinical trials for these patients, so they're waiting for something that we can provide them. I think the key message is that in terms of this field, that even though it's been challenging to identify molecular targets, these are times rapidly changing in terms of our understanding of the epigenetic basis of these specific tumors, and I think we're building models as well. So I think in this particular pediatric brain tumor type, and could be more generalized in terms of all pediatric brain tumor types, that we're developing both new approaches to understand the disease and models, and I think pairing those together will allow us to develop new therapies that we could advance pretty rapidly into clinical trials. So I think even though for some tumor types that may not have agents available, I think our understanding of the disease is rapidly progressing for us to be able to develop those.